Here's the thing. Science doesn't stop just because the news cycle gets chaotic. While the internet argued about AI and politics this year, researchers were steadily making breakthroughs in medicine, ocean exploration, and space astronomy. They were pushing forward in ways that could reshape our picture of life on Earth and far beyond it. These weren't the headlines dominating social media, but they probably should have been. To give you a taste, think new horned dinosaurs reshaping evolution charts, hundreds of previously unknown ocean creatures cataloged, and even James Webb hinting at chemical traces tied to life in distant worlds. Today, we're counting down 23 staggering discoveries you likely didn't hear about. Some are massive, others surprisingly small, but all challenge what we thought was possible. And one of them starts with sharpening the limits of human vision: night vision contact lenses. One of the more unusual ideas researchers are exploring is whether contact lenses themselves could one day allow humans to see in conditions that normally look like total darkness. Instead of relying on bulky goggles or glowing screens, experimental work in material science has suggested it may be possible to build lenses that pick up parts of the light spectrum our eyes cannot naturally detect, specifically infrared. That's the range of light just beyond red, invisible to us but carrying clear information about heat, contrast, and structure. At the moment, this is still laboratory-level research rather than something you can order online. The general concept is that certain nanoparticles and optical materials could be engineered to convert those invisible wavelengths into signals our eyes recognize. A lens built with those components would still appear clear and wearable, but it would essentially act as a small filter that reveals patterns lost in the dark. Nothing like a finished human-ready pair exists yet, but the possibility of embedding this kind of ability into something as thin as a contact lens has kept research groups interested. What could it mean if this idea becomes practical? Think about rescue teams moving through collapsed buildings, smoke-filled spaces, or unlit landscapes. Being able to trace heat signatures without carrying extra gear could save time and lives. Firefighters might see through haze to locate people faster. There's also interest from security fields. Many products and official documents are tagged with markings visible only under specific wavelengths, and a lens that highlights these would let inspectors recognize them without external tools. Medical uses have also been suggested, from tracking subtle temperature gradients on the skin to supporting new forms of imaging. The hurdles here are significant. A material placed directly on the eye has to be perfectly safe, non-irritating, and durable. Any heat buildup could cause harm, so designs must balance sensitivity with comfort. Regulatory testing would be extensive before anything reaches patients or consumers. And beyond safety, there are ethical questions. Who gets access to augmented vision, and how would it change the balance between individuals who can afford it and those who cannot? So for now, functional night vision contacts remain a concept, not a product. Still, the research shows how quickly the boundary between human senses and engineered enhancements is shifting. And while this remains a vision of the future, our next story is about a medical breakthrough that has already changed the present. Cure for sickle cell anemia. For decades, sickle cell anemia has stood out as a disease with little more than temporary fixes. Instead of the standard round, flexible red blood cells, patients inherit cells that twist into crescent shapes. Those stiff cells clog blood vessels, deprive tissues of oxygen, and shorten lives. Most available options could ease pain or manage symptoms, but the illness itself continued unhindered. Today, however, medicine is beginning to explore something far more ambitious: the possibility of reprogramming the body's own biology to correct the defect at its root. Instead of transfusions or drugs borrowed from other conditions, researchers are turning to genetic and cellular approaches that directly modify a patient's blood-forming cells. The central idea is simple but radical. Take a person's own marrow stem cells, alter them to carry fresh instructions, and then return them to build a healthier blood supply. Rather than masking the problem, the treatment attempts to rewrite the instructions that caused it in the first place. The process, still under careful study, is intense. Patients undergo collection of bone marrow stem cells, which are then engineered in controlled laboratory settings. Those modifications allow the altered marrow to produce red blood cells less prone to dangerous sickling. After the patient's existing marrow is reset to make room, the edited cells go back in, where they begin, producing new blood on their own. Over time, corrected cells may outcompete the diseased ones, leaving fewer painful blockages and, in many cases, none at all. Results from early studies suggest this approach offers lasting relief rather than just day-to-day management. What makes this effort especially significant is its impact on communities that have been waiting for solutions. Sickle cell anemia primarily affects people of African descent and other populations historically underserved by medical innovation. For many families, years of hospital stays, organ damage, and fear of sudden complications define their expectations of the disease. A therapy that aims to cure, instead of simply alleviate, signals a shift in that narrative. It demonstrates how advances in gene editing and cellular engineering are beginning to reshape the outlook for inherited conditions once labeled as permanent. That shift raises broader questions for medicine as a whole. If we can precisely target genetic defects in the blood, what else becomes possible? Could entire categories of incurable disease be reframed as solvable problems of code and repair? Those questions are actively being tested, not only in blood disorders, but in other areas where early work suggests new ways of training the body's defenses against some of the toughest challenges known in healthcare. Pancreatic cancer mRNA vaccine. Cancer treatments are also testing new frontiers, and one of the most closely watched experiments involves a potential mRNA vaccine directed at pancreatic cancer. This form of cancer is among the most lethal, with survival rates remaining stubbornly low, even when detected early and treated aggressively. That difficult reality is why the very idea of a vaccine tailored not to a virus, but to a tumor, has raised so much interest within research hospitals and trial centers. The principle behind this effort is to train the immune system to recognize the genetic mistakes carried by a patient's tumor. Instead of treating all patients in exactly the same way, the vaccine would be built using information gathered directly from an individual's tumor cells. mRNA instructions would then signal the immune system to hunt for the same abnormal proteins produced by cancer, creating a line of defense that previously never formed on its own. Because pancreatic cancer is so notorious for slipping past detection and returning even after surgery, reinforcing the immune system could add years of protection if proven to work. Researchers emphasize that this approach is in the early testing stage. Small clinical trials run by groups, including those partnered with BioNTech and Moderna, are monitoring how patients respond, whether the immune signal is strong enough to matter, and, most critically, whether relapse rates remain lower in the long term. The vaccines draw directly on methods refined during the pandemic, but are repurposed here for precise one-patient-at-a-time medicine. Every dose is unique, designed to match a single cancer profile rather than a community of susceptible individuals. So far, findings suggest the immune system can indeed be guided to notice pancreatic tumors more effectively, though the durability of that response will not be clear for years. Doctors are cautious, noting that cancer biology is far more complex than a virus, and progress is likely to come in measured steps rather than sudden leaps. Still, even limited success would mark a turning point in how we think about treating one of medicine's most unforgiving diagnoses. A vaccine designed to intercept cancer before it reemerges represents a striking extension of what's possible when biology can be programmed with precision. And while oncology pushes in that direction, another frontier of lab work is showing how scientists can now build human structures that only used to appear during the very first weeks of development. Lab-grown human backbone, notochord. Scientists have recently managed to recreate one of the most fundamental structures of early human development, the notochord, a core scaffold that guides the formation of the spine. Rather than studying it directly in embryos, which raises both ethical and practical limits, researchers built a lab-based model of this structure using human stem cells. By carefully adjusting the environment surrounding those stem cells, scientists were able to coax them into organizing in ways that mimic the notochord's behavior. Though this is not a fully developed human backbone, the model captures many of the key functions that make the notochord so important. During development, it sends out molecular signals that tell other tissues how to shape themselves into bones, discs, and parts of the nervous system. Having a version that scientists can observe outside the body lets them study those signals in unprecedented detail. One clear application of this research is understanding why certain congenital spine conditions occur. Many deformities arise when those early notochord signals fail or arrive in the wrong sequence, leading to issues with vertebrae or spinal closure during development. Using a lab-based version, researchers can replay those early stages, watching errors unfold and testing whether specific interventions might restore proper signaling. This could help medical science explain conditions like scoliosis or spina bifida and potentially guide the design of therapies that prevent them in the future. The model also holds potential for solving problems that emerge much later in life. After birth, remnants of the notochord persist as the soft material inside intervertebral discs, the cushions that keep vertebrae flexible. These discs weaken with age, and their deterioration is one of the most common sources of chronic back pain worldwide. With a lab-grown notochord, scientists can not only study how discs lose their strength, but also test new ways to restore or even regrow them. Instead of managing pain with surgery or drugs, regenerative treatments built on this research could eventually provide more lasting relief. Significantly, models like these avoid the need to use embryos at all, offering both a scientific and ethical advantage. By sidestepping one of the largest hurdles in developmental biology, researchers can push forward with studies that were nearly impossible before. The promise is not instant, but it points toward a long-term goal, replacing damaged tissues with lab-grown structures that naturally integrate into the body. If early development can be recreated and studied so closely under controlled conditions, it raises a bigger question: what other systems might be powered by this kind of innovation? Some scientists are already exploring possibilities that extend from our cells all the way to the energy that fuels our world. Sunlight reactor for green hydrogen fuel. Imagine a filling station where instead of gasoline or diesel, the pump dispenses fuel created from nothing more than water and sunlight. That possibility has moved from theory to prototype, as scientists have demonstrated devices that use photocatalytic materials to split water into hydrogen and oxygen using only the energy of sunlight. While still experimental, this approach points to a cleaner alternative to fossil fuel-based hydrogen production. At the center of these systems are thin photocatalytic sheets coated with specialized compounds. When sunlight strikes their surface, the incoming photons trigger electrons into action, driving the chemical reaction that separates water molecules. What emerges is hydrogen gas that can be stored, transported, and later used in engines or fuel cells. The contrast with conventional hydrogen production is sharp. Most existing supply is produced from natural gas, releasing significant carbon emissions in the process. By skipping fossil fuels entirely, the sunlight-driven design removes one of hydrogen's biggest drawbacks. Hydrogen itself has always been attractive because of its clean exhaust. Burn it in an engine or run it through a fuel cell and the only byproduct is water vapor. The stumbling block has never been its potential, but the economics. Splitting water with electricity is energy-intensive and costly. A reactor that draws directly from sunlight reduces that expense by eliminating the need for a separate electrical input, potentially changing hydrogen's role from niche fuel to scalable energy carrier. Scaling, however, remains the challenge. To displace even a fraction of fossil fuel use, reactors would need to cover vast areas in arrays, similar to solar farms, steadily generating and storing hydrogen in large volumes. If that hurdle can be crossed, the applications are broad. Powering transport fleets, fueling industries with high energy demands, and providing long-term energy storage to complement solar and wind, which fluctuate with weather and daylight. Storing electricity as hydrogen would make renewables more reliable, with tanks of sunlight-derived fuel available even when nature pauses. Researchers are careful to note this technology is in its early days, but the promise is clear. If costs continue to fall, hydrogen could play a central role in decarbonizing heavy transport, shipping, or grid storage, fields where batteries alone may not be enough. From the global scale of fueling whole economies, researchers are also applying similar ingenuity to the fate of individual species, where breakthroughs in the lab may now determine whether endangered animals still have a future. Panda stem cells. Breeding pandas has been one of the most persistent puzzles in conservation. Even when conditions in captivity are carefully managed, the timing is unforgiving. Females are fertile for only a short window each year, and many mating attempts, natural or assisted, do not result in cubs. With wild populations still hovering at vulnerable levels, researchers have searched for a backup strategy. One of the most intriguing developments has been the ability to reprogram ordinary panda skin cells into stem cells that can, at least in the lab, be nudged toward many different pathways of growth. Stem cells are often called biology's universal raw material because of their flexibility. In controlled conditions, they can become muscle, bone, nerve tissue, or other specialized cells. By generating them from skin, scientists avoid removing embryos or eggs from pandas themselves, making the process less invasive and more sustainable. Though the work is still at the experimental stage, it provides researchers with a renewable supply of material for studying panda biology in ways that weren't previously possible. the conservation implications researchers hope for are significant. In theory, these stem cells could help support reproductive efforts in captivity by providing insights into developmental processes, or one day opening the door to creating embryo-like structures under strict legal and ethical oversight. While these applications remain speculative, the technique itself has already suggested a new avenue that was unavailable when conservation depended only on natural breeding and artificial insemination. Another potential use lies in understanding panda health. Because the animals are rare, direct experimentation has always been difficult. With induced stem cells, scientists can grow panda tissues in the lab and study how they respond to infections or environmental stresses. That kind of research can guide new treatments and help anticipate diseases that might otherwise threaten the population. It also cuts down on the need for invasive procedures in living animals, reducing risk for the species while expanding what scientists can learn. There's also long-term interest in addressing the genetic narrowness of panda populations. Inbreeding remains a serious concern, leaving pandas more vulnerable to illness and environmental change. Stem cell methods might someday form part of a strategy to reintroduce diversity. That said, experts caution this a distant scenario, and careful testing will be required to avoid disturbing natural reproduction cycles. These efforts highlight how conservation now often starts at the cellular level, with scientists exploring how the smallest building blocks of life could safeguard a species. And this focus on cells is not limited to animals. Recent work on human biology has revealed surprising clues about diseases once thought to have no clear explanation. Discovery of lupus cause and potential reversal. Lupus has long stood out as one of the most puzzling autoimmune conditions, marked by an immune system that inexplicably begins to harm the very tissues it is meant to protect. Patients face cycles of fatigue, pain, rashes, and sometimes serious organ complications, often without a clear explanation for why their defenses have turned against them. For years, most treatments have focused on easing those symptoms or broadly suppressing immune activity, which carries its own risks. Now, research is offering a sharper picture of what may sit at the center of this disease, a fundamental misalignment in the immune system itself. The focus has turned to T cells, a family of white blood cells that normally balance attack and repair. In people with lupus, recent studies have found evidence of an unhealthy split. There appear to be too many aggressive T cells that cause damage, and not enough regulatory T cells tasked with calming inflammation and supporting recovery. It is this imbalance that may explain why lupus is both destructive and relentless. Instead of shutting down after the damage is done, the immune system remains on an accelerating track. Tracing this misuse of the immune system led scientists to a powerful signaling protein called interferon. Under normal circumstances, interferon primes the body to resist viruses, rallying defenses at the first sign of infection. In lupus, however, emerging evidence points to interferon as a constant spark that keeps destructive T cells energized while pushing the protective ones into the background. The result is an immune system locked into self-sabotage. This insight has already shifted attention toward interferon as a potential therapeutic target. If scientists can control or quiet its overactivity, there is hope that balance between destructive and regulatory T cells could be restored. Early efforts in this direction are underway, involving drugs designed to influence interferon pathways and bring harmony back to immune signaling. While these treatments remain investigational, the approach represents a move away from blunt suppression toward therapies that may actually correct the underlying process. For patients, the implications could be profound. Instead of facing a lifetime of managing flare-ups, targeted interventions could one day retrain the body to regulate itself more effectively. It's not a cure today, but it is the first real outline of how one might be achieved, changing lupus from a mystery without solutions into a clearer problem with a direct strategy. And just as these immune therapies suggest a shift in how we tackle human disease, scientists working in another field are reimagining how to safeguard life itself, turning to advanced reproductive tools to give a nearly lost species a second chance at survival. Rhino in vitro fertilization, IVF. In September 2023, conservation scientists reached a milestone with rhino reproduction that was once thought impossible. They succeeded in creating a viable embryo through in vitro fertilization and transferring it into a southern white rhino, which resulted in a confirmed pregnancy. For a species group facing constant pressure from habitat loss and poaching, this was not just a technical experiment. It was proof that reproductive technology could provide a new safeguard against extinction. The process itself mirrors the basic steps of human IVF. Eggs are collected from female rhinos, sperm is obtained and preserved from males, and fertilization takes place under controlled laboratory conditions. Only embryos that show promise are then implanted into chosen surrogates. In this case, the surrogate was a healthy southern white female selected for her ability to reliably carry a pregnancy. IVF differs from artificial insemination because it allows scientists to monitor embryo quality before transfer, increasing the likelihood of a successful outcome. The urgency behind this effort lies with the northern white rhino, a subspecies reduced to just 2 living females, neither capable of natural reproduction. By demonstrating success with southern whites, closely related relatives, researchers can refine methods needed to implant northern embryos created in labs. If northern calves can eventually be brought to term by southern surrogates, it could preserve a genetic line otherwise destined to vanish completely. This approach helps solve one of the hardest problems for near-extinct species. There are often too few animals left for natural mating, and those that remain lack the social stability and genetic variation needed to sustain a population. Assisted reproduction breaks through those barriers, making it possible to continue reproduction even when the normal biological system has shut down. Challenges remain. Embryo transfer across subspecies boundaries may not always succeed, and each stage, from egg retrieval to implantation, requires painstaking monitoring. There is also no guarantee that a successful pregnancy leads to a thriving calf, let alone to a self-sustaining breeding population. Still, the southern white pregnancy has been a powerful signal that the conservation toolkit is expanding, moving from theoretical solutions to practical steps with real biological impact. Could this one technique restore a nearly lost subspecies? The jury's still out, but this is the first time the path was proved possible. And as reproductive biology opens unexpected doors, another area of medicine is beginning to show that even devastating genetic disorders once seen as unstoppable may soon meet meaningful resistance. Groundbreaking gene therapy for Huntington's disease. Huntington's disease is among the most difficult inherited conditions known to medicine, and for decades it has carried the weight of inevitability. Unlike many illnesses that come and go, this disorder, caused by a mutation in the Huntington gene, gradually dismantles movement, thinking, and personality. The defective gene tells brain cells to manufacture a toxic protein, which accumulates until neurons in regions key to memory and coordination die off. Once the mutation is passed on, the outcome has historically looked like a slow decline with no reliable way to change course. Researchers are now beginning to test a different strategy, targeting the source of the disease rather than just its consequences. Early clinical trials of gene therapies designed for Huntington's have shown encouraging results, suggesting it may be possible to reduce the amount of harmful Huntington protein produced in the brain. While these studies are ongoing and involve small groups of participants, the idea itself is groundbreaking, an approach that aims to intervene at the genetic level, rather than focusing only on symptom relief. The therapy uses a modified viral vector as a delivery system. It doesn't cause illness, but acts as a carrier, transporting therapeutic instructions into affected brain cells. Once inside, these instructions are designed to silence or reduce the faulty gene's activity. Instead of battling tremors with one drug and mood changes with another, this technique seeks to cut the disease off closer to its origin. It's an attempt to change biology that triggers the damage, offering a more precise layer of control than earlier treatments. Reports from trial participants have added weight to the scientific data. Many patients showed slower decline than what would normally be expected, with improvements observed in areas like speech and motor stability. While such outcomes cannot yet be generalized across the wider Huntington's community, the stability itself is important. A slower rate of change means more time with independence intact, more time for families to share routines without constant new losses, and more years of relative quality living. It's important to recognize that this therapy is at an early stage. Larger studies are needed, long-term safety must be confirmed, and the treatment is not being called a cure. Still, the cautious optimism among clinicians and families speaks volumes. By shifting from managing damage to addressing its root cause, this research offers a chance to reframe how Huntington's disease is understood and how it might one day be controlled. And while breakthroughs in genetics are reshaping life on Earth, some of the most surprising discoveries this year came from looking far beyond it, where astronomers think they may have spotted something the universe has never revealed before. James Webb Space Telescope discovers possible black hole star. Astronomers working with the James Webb Space Telescope have reported data pointing to objects that challenge much of what models predicted about the early universe. While examining some of the most distant regions ever observed, Webb picked up faint red sources of light that looked brighter and more massive than expected for galaxies at that stage of cosmic history. Instead of fitting neatly into categories of young galaxies or stars, their light signatures suggested something unusual was underway. One proposal, still debated, is that these anomalies might represent exotic stages of stellar evolution, objects some have tentatively labeled black hole stars. The concept describes a massive star dense enough for a black hole to form at its core while the outer layers continue to shine. In theory, this would produce an object that glows like a star but channels matter inward to grow a black hole at an accelerated rate. Whether these candidates truly represent that state remains uncertain, but the idea highlights how much our understanding of the first luminous bodies may expand as Webb's data is analyzed. The key advantage Webb brings to this puzzle lies in its infrared vision. Earlier telescopes could not see through the dense fields of cosmic dust that mask so much of the early universe. Webb's instruments, however, can capture stretched-out infrared light, traveling to us across more than 13 billion years. That capability is what allowed astronomers to notice these faint, distant red sources in the first place. Their brightness and apparent size stand out immediately when compared with projections based on standard galaxy and star formation models. Why does this matter? Because astrophysicists have long struggled to explain how supermassive black holes appeared so soon after the Big Bang. Forming one through ordinary stellar collapse should take far longer than the universe seemed to allow. If some early stars both shone outwardly and grew black holes within, they could provide a faster mechanism for producing these giants, which later shaped the growth of entire galaxies. Webb's glimpses of outliers may therefore represent key links in a major cosmic mystery, the jump from small stars to massive black holes that restructured their neighborhoods. For now, astronomers are testing these findings against alternative explanations. The unusual light fingerprints might be misinterpreted signals, or they could reflect the presence of energetic disks of matter swirling into black holes inside young galaxies rather than anything fundamentally new. Either result will reshape our models. If these candidates hold up, researchers may be looking at objects that blend extremes of gravity and brilliance in ways never confirmed before. The lesson is that many of the universe's most striking features remain hidden, waiting for the right tools to bring them forward. And while Webb stares back across billions of light years to detect elusive signals, here on Earth another set of discoveries is unfolding much closer to home, buried beneath some of the thickest ice on the planet. Discovery of 85 active subglacial lakes beneath Antarctica. Beneath almost 4 kilometers of Antarctic ice, researchers have discovered that far more hidden lakes are active than anyone once assumed. Previously, only a few dozen subglacial lakes were known to shift, drain, and refill under the ice sheet. But recent satellite analyses, particularly from the European Space Agency's CRIOS at 2, revealed many more such systems. The findings showed that Antarctica does not simply trap water in frozen stillness. It hosts a dynamic, shifting network of lakes that operate out of sight. These lakes behave like underground reservoirs that fill and empty under enormous pressure. As water moves from one cavity to another, it reduces the friction at the glacier's base, allowing ice above to slide more easily across the bedrock. That sliding action is not just a local curiosity. It directly affects how quickly the ice sheet can flow toward the Southern Ocean. Because Antarctica contains enough ice to raise global seas by many meters, this hidden plumbing system plays a critical role in the future of coastlines worldwide. Models of sea level rise now need to account for the fact that what happens under the ice may speed things up in ways that surface measurements alone cannot reveal. Another layer of intrigue comes from biology. Locked away from sunlight and contact with the atmosphere, some of these lakes may have been sealed for millions of years. Conditions that are cold, dark, and under crushing pressure, yet scientists believe microbial life could survive there, adapted to extremes well outside everyday experience. Discovering such organisms would be significant not only for understanding Earth's resilience, but also for evaluating where life might persist elsewhere in the solar system. Subglacial lakes, isolated and self-contained, are natural analogs for ice-covered oceans on moons like Europa. Reaching these lakes directly remains daunting. Drilling through kilometers of ice without contaminating the environment involves major engineering challenges, and so far most of the evidence comes indirectly. Satellites like CRISPR pick up subtle rises and falls in surface elevation, showing where water collects beneath the sheet and when it drains away. What were once invisible voids can now be mapped and monitored with remarkable precision. Taken together, these findings expand Antarctica's hidden geography and force scientists to sharpen their view of how the planet's largest ice mass actually behaves. If we want to forecast how coastlines may change in decades to come, understanding this shifting underworld is essential. And just as satellites help reveal unseen systems under the ice, another new tool has recently allowed scientists to uncover hidden processes within the human brain itself. New tool for understanding memory formation. The question of how memories take shape inside the brain has long hovered without a clear answer. Scientists have known that new experiences leave physical changes in neurons, but catching that process in action has been almost impossible. Recently, researchers have introduced a tool that provides a clearer view of this puzzle by tracking how proteins are built inside neurons at the precise moment they're needed. At the center of this is dendritic translation, the ability of neurons to manufacture proteins directly in their branching extensions rather than relying only on the cell body. Those proteins serve as reinforcements at synapses, the junctions between neurons, strengthening connections when signals pass through them. You might think of it like carpenters adding braces to a bridge while it's being crossed, ensuring the structure grows sturdier at the exact spots where weight is carried. This localized reinforcement underpins what neuroscientists call synaptic plasticity, the bedrock for turning fleeting experiences into lasting memory. By following protein synthesis in real time, scientists have seen more than just reinforcement happening. They have uncovered numerous regulatory processes that act like fine controls, adjusting how much protein is produced, how long it sticks around, and which connections receive support. Memory, it turns out, isn't a simple system of switches flipping on. It looks more like a set of carefully tuned levers, each one shifting the balance of how an experience gets stored. That complexity helps explain why memories can be vivid and durable in some cases, or fragile and inconsistent in others. Understanding this mechanism opens up clearer paths into medical challenges. In Alzheimer's disease, for instance, neurons lose their ability to sustain those local protein factories. Synapses weaken instead of holding steady, and connections that once supported recall begin to vanish. With this tool, researchers can now see where the breakdown occurs, which has already begun to inform strategies for slowing or interrupting that decline. In principle, therapies could target the specific regulatory steps that falter rather than broadly suppressing symptoms after the damage is already done. On the other side of the equation, there's also the potential to enhance memory by adjusting the very dials this tool highlights. Strengthening local protein synthesis could one day improve retention for people with impairments, or even sharpen healthy memory beyond ordinary limits. That remains a prospect for the future, but the ability to finally watch memory formation unfold represents a turning point in neuroscience. And while researchers draw new maps of how thoughts solidify, other scientists are beginning to notice that cells outside the brain, once considered background players, could be shaping some of medicine's most perplexing battles in ways nobody expected. Sensory neurons promote cancer metastasis. Cancer research has begun to reveal that nerves may play a more active role in disease than once thought. For many years, scientists assumed the nervous system was only a bystander, delivering signals for sensation and movement, but not influencing tumors directly. Recent animal model studies, however, suggest that sensory neurons might help cancer spread by providing chemical instructions that encourage tumor cells to migrate and invade new tissue. Evidence has been strongest in breast cancer models. When researchers observed interactions between tumors and their surrounding nerves, cancer cells were not acting entirely on their own. Instead, they responded to molecular cues released by nearby sensory neurons. Those signals appeared to guide the cell's behavior, telling them when to move, where to infiltrate surrounding tissue, and how to branch out toward distant sites. The outcome was more aggressive metastasis, the stage when cancer escapes its original site and becomes both deadly and difficult to treat. This finding reshapes the way scientists think about the tumor microenvironment. Until now, it was understood to include immune cells, blood vessels, and connective tissue. Nerves were placed outside that conversation, thought to be relatively neutral. The newer evidence suggests they may instead act as partners in progression. one more element in a system that cancer recruits to its advantage. That changes how researchers frame the challenge. Controlling tumors may also mean addressing the body's communication wiring, not just the tumor cells themselves. For treatment, the idea opens a possible new angle. If nerve-to-cancer communication really does help metastasis, some of the molecules that sensory neurons release already have been studied in other conditions, which suggests that adapting drugs to interrupt these pathways might be possible. Such a strategy would not replace surgery, chemotherapy, or targeted gene therapies, but it could reinforce them, cutting off some of cancer's escape routes before tumors spread. The broader implication is that cancer may best be understood not only as a collection of mutant cells, but as a system-level disease intertwined with many normal processes of the body. Signals from the nervous system may be part of why traditional approaches aimed squarely at cancer cells haven't always been enough. New treatments that acknowledge this larger network could someday reshape how oncologists prevent metastasis, the phase that makes cancer hardest to overcome. And as researchers broaden their focus to these hidden lines of communication, another team has uncovered an unexpected defense system inside our own biology, one that can trigger a destructive countermeasure the moment an invader gets through. CRISPR system as a molecular fumigator. CRISPR is best known as a molecular tool for cutting and editing DNA with precision, but not all CRISPR systems behave like tidy scissors. In bacteria, some versions operate with a very different purpose, serving as a last resort defense mechanism when viruses invade. Instead of quietly fixing or trimming sequences, these systems flood an infected cell with destructive molecules, forcing it to shut down before the virus can spread. This response is what microbiologists describe as an abortive infection. Logic is simple. One cell sacrifices itself so the larger community survives. By collapsing the machinery that viruses need to replicate, the doomed cell prevents the invader from multiplying and moving further through the colony. Researchers liken this to a molecular fumigator. Rather than surgical edits, the system fills the compromised environment with toxins, cutting off infection at the source. It is not elegant, but it is efficient. In practice, this operates like an internal self-destruct. Conventional CRISPR tools carry out targeted corrections, but the fumigator mode treats the entire infected cell as hostile territory. Once triggered, the system makes the space uninhabitable, ensuring nothing gets out alive. One vivid way of putting it, it works like a biological smoke bomb, sealing the door and clearing the room in one move. What makes this especially compelling is how it reframes CRISPR itself. For years, the technology's reputation centered on editing, precise cuts and paste instructions that let scientists adjust specific genes. But the fumigator role shows CRISPR can be much more. Not just a maintenance kit for genomes, but a larger defense platform that changes strategy depending on the threat. This dual capacity, precision repair in one setting and total demolition in another, expands how scientists imagine building new therapies. Could this mechanism someday translate into antiviral treatments for humans? Researchers are exploring the idea, but progress remains cautious. The insights from bacteria suggest it might be possible to program human cells with a similar kind of emergency shutdown, preventing viruses from taking hold. That said, adaptation for medicine is still speculative, and the challenges of safety and control are substantial. For now, the fumigator role is best understood as inspiration, an example of how evolution has already solved problems we are only beginning to confront. Seeing CRISPR's versatility this way reminds us that biology often hides multiple layers of function in the same system. And just as CRISPR reveals unexpected power in the way organisms defend themselves, another recently developed tool is beginning to reveal hidden conversations between cells themselves, making visible a social network inside living tissues that was previously invisible. Lipstick, Universal Cell-to-Cell Interaction Mapping Tool. One of the most ambitious tools now emerging in biology is a tagging system designed to show how cells connect with each other in real time. For decades, researchers have known that the organization of tissues and organs depends on direct contact between cells, yet those interactions were almost impossible to trace as they occurred. Indirect signals, chemical readouts, or delayed markers left scientists guessing at who actually touched whom. The new approach changes that cells can now mark their partners the moment contact happens, leaving behind a record of the interaction and creating maps of cellular connections as they unfold. What makes this powerful is the ability to observe living tissue with clarity that simply wasn't possible before. Instead of only measuring the aftermath of signals floating through the bloodstream or between neurons, researchers can now capture the exact pairings behind them. It is the difference between inferring a city's traffic system by measuring fumes in the air versus having direct logs of which cars cross which intersections at what time. In biology, that kind of tracking lets scientists visualize the networks that guide development, healing, and defense with far more precision than before. The payoff becomes especially clear in the study of immunity. The immune system works like a layered chain of command. One cell detects a threat, another raises alarm signals, and others swarm in to attack or calm the response. Until recently, much of this order had to be pieced together indirectly. With tagging tools, scientists can finally watch specific immune cells attach to their exact targets during infection and confirm how that chain of command is carried out step by step. This closes a gap that left researchers speculating for years about how immune coordination really begins. Development is another area likely to benefit. As embryos form, certain cells broadcast molecular instructions that guide others into specialized roles: bones, muscles, nerves, or organs. Seeing these contacts directly allows scientists to trace the handoffs that shape entire tissues. If an error occurs in the choreography, the tool could reveal where the misstep begins and open possibilities for correcting it before development goes off track. Even in adult tissues, the method is already pointing to applications in conditions like cancer or autoimmune disease, where miscommunication drives much of the damage. By showing exactly which cells pass the wrong messages, future therapies could target the interaction points rather than trying to shut down entire systems at once. What these maps ultimately provide is a new way to decode the hidden patterns of biology. And as researchers sharpen their view of how cells communicate with their immediate neighbors, attention is also turning to the brain, where discovering a new kind of neuron has begun to answer a very different question: why some bodies feel full sooner than others. New neurons regulating appetite. Researchers are now reporting the discovery of a previously unknown class of neurons that play a central role in appetite regulation. These cells act as accelerators for satiety, sending signals of fullness at moments when other systems still lean toward hunger. By responding so quickly, they introduce a layer of control not previously accounted for in the brain's appetite circuitry. It's an emerging finding that helps complete the map of how the body manages the balance between hunger and satiety. What sets these neurons apart is their speed. Normally, appetite is a tug-of-war between hormones that push food-seeking and those that suppress it, with signals traveling through multiple pathways before reaching balance. These newly identified neurons work faster, overriding intense hunger drives before they gain momentum. In effect, they look like a rapid response mechanism designed to quiet cravings, acting well ahead of slower hormonal feedback. For scientists, that acceleration explains why some bodies reach a sense of enough sooner than others. This discovery strengthens the understanding of satiety as a biological state rather than a matter of willpower alone. Appetite control involves layers of receptors, neurotransmitters, and feedback loops, and the addition of these neurons helps fill a missing gap in that circuitry. Where earlier models explained hunger suppression as gradual and hormone-heavy, this finding shows there's also a built-in fast lane, adding detail to a system that already shapes daily energy balance so powerfully. Possible medical applications are attracting attention, especially around obesity and metabolic disease. Traditional diets often fail because they confront biology with rules rather than altering its signals. If these neurons can be influenced directly, therapies might encourage the body itself to limit intake without depending entirely on restraint. That could translate to treatments where hunger fades biologically instead of by effort alone. While any such intervention remains speculative, it offers a glimpse of how appetite research may evolve once detailed neural control points are included in the plan. The more immediate value comes in redefining how eating behavior is framed. By showing that satiety is enforced via neuron activity, not just hormones or psychology, researchers can pursue more precise targets for intervention. Drugs that modulate these specific cells could someday offer weight control strategies with fewer side effects than blanket appetite suppressants. While far from clinical reality, it underscores how appetite research is moving toward understanding fine-tuned circuits, not just broad systems. The broader message is that even states as familiar as hunger follow structured wiring hidden in our biology. But those circuits don't only matter in the moment. They connect to larger schedules that guide how the body changes across an entire lifetime. Aging as a developmental stage. Most people picture aging as a long sequence of wear and tear, joints grinding down, skin losing elasticity, organs weakening at different paces. It's a familiar narrative, but newer studies suggest there may be more structure behind it than simple decline. Recent single-cell sequencing work point to coordinated changes across tissues, leading some researchers to propose that aging may carry elements of a programmed stage of development, more like puberty than mechanical breakdown. This remains debated, but the evidence is intriguing enough to reshape how scientists think about what aging actually represents. The strongest support comes from microscopic analysis, one cell at a time. By applying sequencing tools to organisms at different ages, researchers noticed that cell populations were not drifting randomly. Instead, specific shifts appeared across multiple organs in synchrony. Liver, muscle, and immune tissues all moved through similar transitions, as if they were responding to the same underlying signal. That kind of system-wide choreography fits less with wear and tear and more with a biological schedule kicking into motion. If that interpretation is accurate, then aging is not only about damage building up, but also about a timeline coded into our cells. And like any other timeline, it might contain checkpoints that could hypothetically be adjusted. Identifying molecular switches or signaling pathways behind these transitions might allow researchers in the future to influence when the body enters certain phases, holding the door open for longer stretches of resilient health. The concept shifts the medical challenge from fixing problems after they accumulate to potentially altering when those problems are even allowed to begin. This new way of framing aging also changes how we interpret everyday advice about health. Exercise, diet, and stress reduction may not simply extend life by slowing damage. They could actively reshape how these molecular programs operate, delaying or softening the body's internal schedule. Experimental molecules are already being studied for their ability to temper such processes, offering the possibility of extending health span, the years of life spent in good physical condition, even before lifespan itself is addressed. There's a speculative but thought-provoking angle here. If our cells are following preloaded instructions, it suggests our bodies might one day offer us the biological equivalent of software settings, not to stop aging completely, but to pause or reschedule parts of it. Would you want the start of aging delayed if science found a way to do it safely? Tell us in the comments what you'd do with an extra decade of healthy energy. Ideas like this push researchers to reconsider not only how bodies change, but also how evolution may have shaped those changes into predictable stages. And with so many mysteries still open, science is also racing ahead in very different territory, where solving problems of scale and control requires tools built not inside cells, but out of atoms themselves. Record-breaking neutral atom qubit array. Quantum computing has been inching along for years with steady but modest increases in capability. Yet recently researchers reported a leak that puts the field on a new scale. Neutral atom qubit systems, once limited to small test runs, are now being assembled into record-sized arrays that hold thousands of qubits in one coordinated platform. For context, most experimental quantum computers still operate with a few hundred qubits, and even stabilizing those is notoriously fragile. Demonstrating systems an order of magnitude larger, where qubits hold their quantum states in sync, signals real progress towards something practical rather than only experimental. The crucial advance here is not just raw numbers, but the ability to keep information stable. Quantum bits must remain in superposition, or simultaneously represent multiple states, long enough to do useful computation. Neutral atoms naturally resist outside interference better than solid-state qubits, making them less prone to errors caused by stray fields or thermal noise. That stability has allowed researchers to preserve coherence across these larger arrays, addressing one of the toughest challenges in scaling up quantum machines. In simplified terms, it's like moving from a handful of independent radio transmitters to an entire coordinated global network. The signal becomes powerful enough to carry complex messages reliably. Error correction has always been the stumbling block for quantum systems, as tiny disturbances can easily collapse states. The fact that neutral atom platforms are showing lower error rates across larger arrays makes the case that scalable, fault-tolerant computers may not be theoretical much longer. This doesn't mean the technology is ready to break codes or model pharmaceuticals tomorrow, but it marks a concrete step toward the kind of hardware capable of those feats. If developed further, the applications are immense. Secure communications could be challenged as quantum systems test existing cryptographic standards while new quantum-resistant codes are created in response. Artificial intelligence could gain a boost from machines, able to explore vast spaces of solutions in parallel, compressing simulations that take supercomputers days into hours. In chemistry and materials science, systems beyond classical reach, complex molecules and exotic quantum states, could finally be mapped with precision. Each step toward stability and scale moves the technology from theoretical promise to practical exploration. Seeing experiments hit this kind of record scale is a reminder that science fiction hardware steadily becomes less fictional. Neutral atoms, used as building blocks, are showing they can be assembled into architectures that hold steady long enough to matter. Progress is still cautious, but the distance to usable machines shortens almost every year. And while quantum engineers focus on making arrays of atoms compute, another group of researchers is taking a very different approach, designing a small wearable patch that uses sensors and automation to accelerate how wounds heal. Wearable device for accelerated wound recovery, AHEAL. What if wound care could be made intelligent, adjusting itself in real time instead of relying only on occasional checkups? That idea sits behind experimental prototypes like AHEAL, a wearable system designed to treat injuries while also tracking how they recover. These devices bring together sensors, AI analysis, and precise micro-treatments in a patch small enough to rest directly on the skin, an approach that shifts wound dressings from passive coverings to active medical tools. At the core is continuous monitoring. Built-in sensors measure moisture, tissue health, and levels of inflammation as the wound changes across hours and days. Instead of waiting for a nurse or doctor to remove a bandage and check progress, the patch builds a live profile of recovery in real time. That information feeds into algorithms trained on datasets of how wounds normally heal at different stages. The AI can then suggest or even carry out small interventions, aligning treatment closely to what the body needs at that moment. The interventions themselves are where the device goes beyond observation. Microdelivery systems inside the patch release medication precisely when an infection risk spikes or healing stalls. Electrical stimulation, already known to promote tissue growth, is another option the patch can apply, fine-tuned automatically based on feedback from the sensors. Instead of a fixed schedule, treatment adapts hour by hour, optimizing the healing environment as conditions change. The main goal is practical: reduce complications and shorten hospital stays. Burns, ulcers, and surgical wounds that normally linger and draw repeat visits could heal more quickly when managed this way. Because infections are caught at an earlier stage, fewer patients would need antibiotics delivered through long courses or risk hospitalization from wider complications. By combining constant oversight with targeted therapy, the device acts like a round-the-clock wound manager rather than a piece of cloth meant only to protect. While lab demonstrations and early trials suggest meaningful gains in recovery rates, the technology still needs larger studies to prove its advantages over standard care. Cost, usability, and safety will all be critical before broad adoption. But the idea of making wound care responsive, not static, could cut healthcare expenses by easing patient loads and lowering readmissions, all while bringing relief more quickly. Technology like this shows how medical tools are beginning to merge with intelligent monitoring in ways that feel almost futuristic. And in another field altogether, ideas once confined to abstract mathematics are now being observed directly, showing that even the hidden geometry of quantum physics can take shape in the lab. Observation of hidden quantum geometry in materials. One of the most striking examples of this shift comes from materials science, where researchers are now reporting emerging experimental evidence of something called quantum geometry, also known as the quantum metric. For decades, this concept existed mainly in theory, a set of equations describing how the wave functions of electrons are arranged within a solid. But recent measurements show that this hidden structure is not just a mathematical abstraction. It actively shapes how electrons travel through real materials. Quantum geometry describes the curvature of electronic wave functions inside a crystal lattice. Instead of electrons following clean, straight lines, their paths bend and adjust according to this invisible geometry. Until recently, these effects were inferred indirectly from models. Now tools such as angle-resolved, photoemission spectroscopy, and scanning tunneling microscopy have begun to reveal signatures that match the predicted maps. Electron behavior that once looked puzzling, sudden changes in conductivity or unexpected resistance, can now be explained by the influence of this geometric framework. The stakes are significant because conductivity in materials is not controlled just by impurities or temperature shifts, but also by this deeper structure. In superconductors, for example, electron pairs gain stability partly due to the quantum geometry that guides their motion. Early indications suggest this hidden property may help explain why certain materials achieve superconductivity at higher temperatures than theory allows. For engineers, this offers a new lever to control electron flow, with possible applications in ultra-fast computing systems where every fraction of resistance slows performance. Beyond superconductivity, the same principle may reshape how we approach renewable energy technologies. Devices that convert sunlight or heat into electricity depend on transferring energy efficiently between photons, electrons, and absorbed heat. If quantum geometry controls the routes electrons can take, then tuning that geometry could make such transfers faster and less wasteful. This could lead to devices that harvest light or thermal energy with greater efficiency, pushing renewable systems closer to their theoretical limits. Imagine electrons not as particles drifting through empty space, but as runners navigating a track whose curves can be redrawn at will, a track that determines how fast and far they can carry energy. Perhaps the most important outcome is that quantum geometry unites puzzles once studied separately: conductivity, superconductivity, and topological effects. By offering a shared framework, it gives scientists a unified way to interpret electron motion and a new set of design principles for building advanced materials. What used to sit inside abstract mathematics now promises practical tools for future technologies. As with many breakthroughs, this one changes how we see structure itself, not only in invisible lattices inside crystals, but in the physical forms of life uncovered here on Earth, where paleontologists recently introduced a predator that adds a surprising twist to our picture of ancient ecosystems. New carnivorous dinosaur species discovered in Argentina. Paleontologists in Argentina have described a brand new carnivorous dinosaur, a discovery that highlights just how varied predator evolution was in the late Cretaceous. Named Shachysaurus nico, this animal was found in fossil beds that once formed part of Gondwana. Its remains show a mix of traits not seen in the dominant theropods of the time, breaking patterns in skull structure and dentition that scientists once thought defined the limits of predatory design. Far from being a footnote in the dinosaur record, Shachysaurus forces a fresh look at how meat-eaters adapted to their landscapes in South America. The skull and teeth provide the most striking clues. Instead of the broad, crushing jaws of tyrannosaurs or the blade-like slicing tools of dromaeosaurs, Shachysaurus carried a narrower skull, paired with teeth arranged in a way that doesn't match known predatory models. This suggests it hunted differently from its more familiar cousins, perhaps targeting prey that required neither bone-splintering power nor knife-edge precision. That single feature alone reshapes the ecological picture, inserting a predator with strategies unaccounted for in previous reconstructions of late Cretaceous food webs. From an evolutionary perspective, the surprise lies in timing. Models have long suggested that major predatory strategies had already stabilized by this point in Earth's history. Yet here is evidence of a lineage carving out a new role, independent of the better-known hunting approaches. It's an indication that predator diversity didn't taper off toward the end of the Cretaceous, but continued branching, refining new techniques under local pressures. In South America's semi-isolated landscapes, evolution seems to have experimented more freely than paleontologists once assumed. The regional context reinforces that point. Gondwana's southern ecosystems produce creatures with traits absent elsewhere, and Shachysaurus appears to have evolved in these unique conditions to occupy a specialized role. Rather than directly competing with bone crushers or pack hunters, it likely exploited prey types or strategies of its own, expanding the boundaries of what carnivores could do. Each discovery like this shows that classic images of dinosaur predators leave out a wealth of variation that once thrived on the continent. Culturally, it's a reminder that the dinosaur family tree keeps shifting. Just when textbooks appear complete, a new fossil rearranges timelines and behaviors. Each new predator rewrites the rules of the ancient food web, meaning our textbook dino pictures keep changing. Finds like this are still hidden, waiting to be uncovered in environments where researchers least expect it. And the same principle holds true today, not only in fossil beds, but also in the deepest corners of the planet, where life persists under conditions once considered uninhabitable. Deep-sea researchers find thriving ecosystems in Earth's most extreme ocean trenches. At the base of the planet's deepest ocean trenches, places where pressure is thousands of times greater than at the surface, scientists once assumed almost nothing could survive. But expeditions have shown that many of these dark environments are more dynamic than expected, with microbial communities and animals adapted to conditions that will crush ordinary life. Far from empty, trenches such as the Mariana, Tonga, and Atacama contain networks of organisms that reveal surprising adaptations to pressure, chemistry, and isolation over geologic time. The most striking discoveries are microbes that resist conditions that should destroy delicate biological machinery. At depths greater than Mount Everest's height, proteins would normally unravel and enzymes fail, yet these microbes have evolved special molecular structures to stabilize themselves. Their survival strategies illustrate not just resilience, but unique solutions to life under extremes, rewriting basic assumptions about how cells function when pushed to their limits. Instead of relying on sunlight, these ecosystems begin with energy produced through chemical reactions between seawater and the deep ocean floor. Nutrients released from rocks provide fuel for microorganisms, which in turn support larger organisms. This creates entire food webs in places that never see the sun. Astrobiologists often point to this as an analogy for how life could persist in oceans hidden under the ice of worlds like Europa, where no light penetrates, but chemistry may provide the same foundation for survival. Explorers have also found creatures that look like prototypes for alien adaptation. In 2024, researchers working in the Atacama Trench identified Dulcibella comanchica, a 4-centimeter amphipod with raptorial limbs for seizing prey. Other finds include a glowing sea slug, Bathydivius caudactylus, which detaches a luminous tail when threatened, and giant tubeworms with no stomachs that survive entirely on partnerships with specialized bacteria near hydrothermal vents. These organisms demonstrate that survival in the deep ocean is not about stretching ordinary biology a little further. It often requires entirely novel strategies. The scale of biodiversity is still being uncovered. The Ocean Census Project recently announced 866 new marine species discovered across global expeditions, ranging from deep water corals to previously unknown types of shrimp and fish. Schmidt Ocean Institute expeditions added to this tally with surveys of seamounts like Salas Y. Gomez Ridge, documenting 160 species not previously recorded there, including dozens new to science. Such results suggest that much of Earth's biodiversity remains undescribed, hidden in zones we once thought sterile. The lesson is simple but profound. When scientists look in places dismissed as empty, they keep finding life arranged in unexpected ways. And that same principle now extends beyond Earth, where astronomers scanning the skies are beginning to detect fundamental building blocks of biology embedded in distant galaxies. Astronomers detect organic molecules in distant galaxies, suggesting widespread life potential. Astronomers observing galaxies billions of light years away have found chemical fingerprints that point to complex carbon-bearing molecules, the same family of compounds that form the basis of biology on Earth. What makes this striking is not the presence of carbon itself, but the confirmation that organic building blocks appear far beyond our solar system. These kinds of detections suggest that the raw ingredients for life are common across the universe, not isolated features of our immediate cosmic neighborhood. The discovery relied on radio observatories like the Atacama Large Millimeter Submillimeter Array and the Very Large Array, which specialize in reading faint molecular emissions. Molecules in space release energy at characteristic frequencies, acting like signatures readable across cosmic distances. When astronomers examined the spectrum from this remote galaxy, the emission lines align precisely with organic compounds. That alignment, verified across multiple instruments, provides a robust chemical record of a galaxy too far to probe directly by any other means. On Earth, carbon's strength lies in its flexibility. It can form long chains, rings, and stable backbones for molecules like DNA and proteins. Seeing the same carbon-based compounds in a galaxy separated from us by billions of years of cosmic time is powerful evidence that these same molecular frameworks are not exceptional to Earth. Instead, they appear as part of a universal chemical toolkit written into the cosmos itself. Because the implications are so large, the results were carefully scrutinized. Astronomers ruled out possible noise or interference from dust clouds and ensured that the emission signatures were consistent across different detections. In astrobiology, where the risk of overinterpretation is high, Researchers stress that confirming building blocks does not equate to confirming life. Molecules alone do not make biology, but they set the stage for it. By clearing this step with rigorous checks, astronomers can confidently say these compounds exist far beyond our corner of space. The broader significance is statistical. If the ingredients that underpin biology are found in galaxies scattered throughout the universe, then the odds that life assembled itself elsewhere rise sharply. Some scientists argue this reflects conditions seeded by cosmic dust, carrying organics across stellar nurseries and embedding them into new planetary systems. That view reframes life not as an anomaly, but as one possible outcome of widespread chemistry. The takeaway is cautious but profound. Building blocks are not life, yet their abundance makes life less likely to be rare. If the ingredients are everywhere, how likely is it that life arose elsewhere? That question lingers as one of the most powerful reminders of what science uncovered this year, setting the stage for a final reflection on what all of these discoveries revealed. From shifting our view of ancient predators to uncovering new life under Antarctic ice and tracing signals from the early universe, these discoveries highlight how science continuously redraws our picture of the world. They remind us that much of nature's complexity often lies unreported, waiting for closer attention. The value in these findings is not only in medical hope or cosmic insight, it's in how they expand the boundaries of what we thought possible across Earth and space. Which of these stories surprised you the most? Share your thoughts in the comments and subscribe for more overlooked science worth noticing.